now. Okay. So I think I will start with introducing you, Rose, and talking about this initiative that the recruiting working group we have. So we decided to start this open meeting to the general community about some topic related with diversity, equity, and inclusion. So the first one of this series is with Rose, that she's actually one member of the recruiting working group, but she has done an amazing work about diversity and inclusion in plant science. But also there will be one soon in September about the 89 initiative. We are working on that. And so we will have somebody from that initiative to join us and talk about their work and what we can do and what are the opportunity there. So let me introduce Rose. So Rose is a postdoctoral research fellow in NSF Plant Genome Program. She completed her bachelor degree at Montana State University and her PhD from the University of Kentucky in 2019 under the guidance of Dr. Nicholas McLachie, I hope I did good. Currently, Rose split her time between Michigan State University, where she works with Dr. Bob Van Boren, and the University of Cape Town, where she works with Professor Jill Farrant. Her work centers around understanding adaptation to extreme water scarcity with a focus on arid biomes in Southern Africa. She uses both ecological and genomic approaches to investigate the mechanism of desiccation tolerance in resurrection plants. Rose is passionate about inclusivity, open science, and decolonizing botany. So thank you, Rose. I also have a question for you. Do you prefer people interrupting you for question, or do you want people to have question at the end?
Yeah, I was actually going to say something about that. I'm happy to have people interrupt with questions if you want, or even type them into the chat. It seems like we have a small-ish group, so if this evolves into a conversation, that would be perfectly fine with me. I'm happy to have it be pretty casual.
Great, thank you. So it's all yours.
All right. Thank you for the introduction, Anarita, and for the invitation to come here and share some of our recent work and have the opportunity to talk to this community. So yeah, I'm going to talk today about a couple of our recent projects where we tried to understand sort of the state of the field in plant science with a focus on issues of representation and participation and equality within the discipline. And it's no surprise that we found some inequalities based on this title. But I wanted to start out with just a little bit, a little bit of background about me, just so you guys kind of know where I'm coming from. So I grew up in New Hampshire, and I spent most of my teenage and early 20s working on small-scale vegetable farms. And so this gave me my interest and love for plants and an appreciation for plant production systems and an interest in food security and food production. And then during my 20s, I had the opportunity to travel pretty extensively. Some of this was for school and the research projects I was involved in at the time, but really the bulk of these travels were centered around exploring my rock climbing hobby. And so these experiences were, very fun and enriching, but one of the things that really struck me in all of these travels, both within the United States and abroad, was just how widespread and devastating the effects of drought could be. And so this sparked my interest in returning to plant science and focusing on understanding adaptations to drought. So this brought me, as I said, back to plant science, and I started a PhD working on desiccation tolerance, which is just what it sounds like. It's the ability to survive desiccation, and this is found in a unique group of plants called resurrection plants. And I'm going to show this little video here on the bottom right of a resurrection plant going from a completely desiccated state and to rehydrating and turning green again in just the course of 48 hours. So this is a pretty fascinating adaptation, and it's found in many diverse species shown here in the upper right. So desiccation tolerance has re-evolved multiple times within the plant lineage. And it's not just me, but others also think that these plants are fascinating and they may hold key insights into how we could engineer systems to survive without water. So that's like the really short and quick of what my empirical research is focused on. But this work brought me to South Africa because there's an amazing richness and diversity of resurrection plants found in South Africa. As you guys probably know, South Africa has some very arid habitats. And so there's many plants, resurrection plants, that are adapted to thrive in these biomes. And so over the past 4 years or so, I've been splitting my time between the United States and South Africa, working on characterizing the ecology and the molecular biology of these plants. So this has been really fun and exciting research, but it has also caused me to reflect on global dynamics within science and some of what seemed to me like really obvious disparities between the resources and facilities that I was exposed to in the United States versus South Africa. So that time spent in South Africa and the reflections of working there, coupled with COVID and being separated from my field sites and the lab, I started thinking about how to quantify some of these inequities that I thought I was observing in science. And so we set out to do a couple meta-analyses to try to understand and identify some of the inequities or disparities that exist within the field. Yeah, so I wanted to tell the story today of these two different projects, both of which address similar questions, but are based on different input data sets or databases. And while they find many similar conclusions in both of these studies, there are some unique differences. So I'm going to tell each of these stories in sequence, and I'm going to begin by talking about a paper that we put out in 2021 that looked at progress and patterns within the field of plant genomics. And for this project, we surveyed about 800 different plant genome assemblies, and this is representative of all of the species at the time that had had their genome sequenced and assembled. So yeah, we were interested initially in just understanding the progress and the patterns within the field of plant genomics. So one of the first things we looked at were how many genomes had been sequenced and assembled, and how the quality of those resources differed. So what I'm showing you here, each one of these dots represents a genome assembly for a different species, and they're plotted over time and in relation to their contig N50, which is basically just a measure of how continuously assembled the genome is, or really like how high quality it is. So genomes with higher contig N50 are higher quality resources. And what we found was that the field of plant genomics has grown rapidly over the past decade, and far more genomes are sequenced and assembled today than just 5 or 10 years ago. Not only are more genomes sequenced and assembled, but the overall quality of those resources is generally improved. We attributed this primarily to the increasing use of long read sequencing technologies and genome assemblies, which are these dots colored here in blue. And as you can see, on average, any assembly that incorporated long reads tended to be more contiguous than those that were just based on short read sequencing technologies. So plant genomics is growing really fast, and the overall quality of the genomes are increasing. But we also wanted to understand how evenly we've sampled the biodiversity within the plant kingdom. So in order to do this, we tried to look at the number of genome assemblies that were available for each order of land plants. So that's what I'm showing you here on the left is the land plant phylogeny at the order level. And these gray bars represent the number of genome assemblies that were available within that order. And we compared that to these blue bars, which represent the number of genome assemblies we'd expect to see if sequencing effort was equally distributed relative to the species richness of that order. And so we identified a number of statistically over and underrepresented groups of land plants. There were some groups that had strikingly few genome assemblies. The ferns are a good example. This is probably for a couple reasons. One, fern genomes are notoriously large and polyploid, so that may have been a barrier. But also ferns don't have a ton of economic and agricultural relevance. Overall, the lineages of land plants that had an overrepresentation of genome assemblies were all economically and agriculturally important groups of plants. So this included brassicales, solanales, cucurbitales, poaceae. You'll notice that these are all of the staple crops and economically important plants. In contrast, the underrepresented lineages were typically speciose and ecologically diverse groups of plants. So there's some big taxonomic gaps in this field, and it's not to say that it hasn't been practical and beneficial to study agriculturally and economically important plants. In fact, this has led to numerous scientific and agricultural breakthroughs and innovations. However, it does seem to have come at the cost of exploring some of the more biodiverse or regionally important plants. And we think this is somewhat concerning because those biodiverse and wild species are the ones that are most likely to be threatened by extinction. And they also certainly contain a wide chemical diversity with potential applications for humans. We looked at a number of other metrics about these genomes that I probably won't go into too much detail here, but we looked at how assembly length and ploidy varied across these resources and orders of land plants, as well as the Conte N50 and the percentage of USCOs or universal single copy orthologs that we found in these genomes. And in general, we found that, there was a lot of variability in the quality of publicly available genomes, and that diploid species were overrepresented, which makes sense because up until recently it's been, well, and even currently, it's quite difficult to assemble high quality polyploid genomes. So based on this first set of analyses, we made a couple recommendations. The first of which is to embrace long-read sequencing technologies whenever possible to generate new assemblies. Given the massive disparity in the resulting quality of the genome assembly, we think that there's no reason not to use long-read sequences if you have the ability. And secondly, we'd urge researchers to expand the taxonomic scope and the domestication status of the plants that we're studying and sequencing. And that in particular, we might think about focusing on generating assemblies for clades that have none, for wild and biodiverse species, as well as for complex and polyploid plants. Okay, so at this point, we felt like we had a pretty good handle on what species people were working with and the quality of the genomic resources. And so the next question we wanted to investigate was to understand where in the globe plant genome sequencing research is happening. And so in order to assess this, we looked at the institution that submitted the genome assembly to NCBI or the other database, and we assigned the genome to the location of that institution. And what we found was that plant genome sequencing is dominated by the United States, China, and Central and Western Europe, with over 3/4 of genome assemblies having been led by a team in one of these three areas. There are other regions of the globe that have notably smaller footprints in plant genome sequencing. And we couldn't help but notice that the pattern of where plant genomes have been sequenced and assembled overlays pretty neatly or closely with the global distribution of wealth. We find that in general, plant genomes have been sequenced and assembled in regions that are relatively affluent. China is certainly a notable exception here, but China has made very strategic investments into science and plant science in particular. So it's not a big surprise, I don't think, to any of us that genome sequencing is generally carried out in affluent regions of the world. It's not a particularly cheap science, but it is concerning, and we think that it's important at this point to acknowledge that the global distribution of wealth, while it's affecting plant genome sequencing and other disciplines or fields within science, has been established in a large part due to the colonial history. And I want to take this opportunity to just segue a little bit to say that it wasn't a huge leap for us to say, oh, these patterns of wealth distribution and plant genome sequencing might be related to colonialism, because there's a long history of colonialism in plant science. And in fact, scientists and botanists or naturalists were among some of the first people to venture to colonial territories and on these early exploratory trips. And in fact, scientific curiosity motivated a lot of early colonial expeditions. On these trips, the naturalists and botanists would often catalog and collect seeds and specimens of species. They would rename them, often with European surnames, and transport them back to facilities and botanical gardens in the global north and in Europe. And to this day, some of our most extensive and well-curated collections of seeds and plant specimens are housed in the global north, limiting access to these resources for people living elsewhere in the world. I thought that this figure is from a 2021 paper by Trisos et al. Is a really good example of this. So what they did is they cataloged the number of bird species that had been named with a European surname. And what you'll see is that the highest density of these European surnames are not in fact in Europe, where we might expect, but they're in these colonial territories. And so this just shows how pervasive this sort of renaming of biodiversity was. Taken together, all of this led to a disproportionate accumulation of botanical wealth and scientific prestige in the global north, and this is continuing to have effects in our science today. So with that being said, we wanted to dig a little bit deeper into our data set to try to understand if some neo-colonial practices were occurring within plant genomics. In order to understand this, we wanted to look at how many of the species that had genome assemblies, how many of those assemblies and projects had been led by teams of researchers working within the native range of that species, versus projects that had been led by teams working abroad, where perhaps the scientists have gone in and collected that biodiversity, brought it back home, and conducted their research on it. So in order to get at that, we looked at the native range of all of the crop species in our database, and we compared this to the teams that had led their genome sequencing and assembly. And what I'm showing you here, each one of these circles represents the number of crop species that are native to that continent. And then the arrows emanating away are scaled by the number and represent teams where those species were sequenced off continent. So you can see there's actually been a fair amount of sort of exchange of germplasm across the globe, with different people sequencing the genomes of species that are not necessarily native to their continent. And if we look sort of across the global north and Asia, we see there's been some reciprocal exchange of germplasm. Excuse me. But when it comes to South America and Africa, we found that all of the crop species that were native to these planets have been sequenced off continent. And so this is a little bit concerning, I think, for a couple of reasons. That there's a lot of reasons, but the ones that stand out to me are that we're likely missing out on the insight and ecological knowledge and wisdom of communities in these regions where the plants are native to. And secondarily, if foreign teams are leading the science on these species, it's very probable that we're prioritizing different things than are relevant to the people working in the native region. For example, a foreign team may come in and sequence a cultivar or a landrace of a species that is actually not relevant to farmers who are producing it in the region. They perhaps grow a different cultivar. So this was a bit concerning, but we thought, well, hey, let's do some more due diligence, because perhaps there's really rich collaborations going on here that include people from the native range of these species. So we looked at all the co-authors on these papers, and we found that for all the papers that were sequenced off continent, these are all the papers sequenced off continent by these 3 different continents. This really only happened in Asia, Europe, and North America. Only about 15 to 25% of those papers included a single collaborator from the native range of the plant. So we think that this is concerning, and it is reminiscent of helicopter science or parachute science. And it suggests that as a field, we could probably do more to engage collaborators in our projects, especially when we're studying species that are not native to where we're located. And this is certainly something that's very relevant to me. I hope it's relevant to other people in the room, but as I've been working with biodiversity from another country, I've had to think really carefully about what is ethical when it comes to studying these plants, and how can we really meaningfully include people who have knowledge of those plants. Okay, so that's the summary of the genome paper. And so we'd found what I thought were some pretty concerning patterns, but I was curious to build on this and to try to understand if this was a phenomena that was just occurring within plant genomics, or if it could be generalized to plant science more broadly. So we set out to conduct another meta-analysis, but this time was significantly larger scale. So for this project, we surveyed almost 300,000 papers published within plant science across the past 20 years. And we sourced these papers from 125 different journals that spanned impact factor, subspecialty, as best we could, the location of the publishers and the location of the journal. And so we think, while we certainly didn't come up with a comprehensive set of all plant science papers, we have a representative set of plant science papers. So we took these 300,000 papers and we did a whole bunch of data science to pull out metrics and to summarize the patterns within the literature. The first thing that we looked at was the global distribution of authors. So we looked at all the authors across these 300,000 papers, and we plotted this density map of where authors are located. And this looks, well, it's in a different orientation than the genome papers, but this looks like a very similar pattern to what we saw with plant genomics. More than 70% of plant science authors were based in China, in Central and Western Europe, and in the United States. There are other regions of the globe that certainly have notable footprints in the plant science literature, including India, Brazil, parts of Australia. But there are other regions, Central Africa and parts of South America and Central Asia, that have virtually no footprint in the plant science literature. Again, I said this before with the other project, but I think this is concerning because as a scientific discipline, this means we're missing out on the wisdom and the insight of those people. And again, we may not have the same priorities as local communities and rural communities within these regions. And so there may be a disconnect between the research and what the application could be in these regions of the world. And this kind of dynamic can perpetuate inequalities. Okay, so in this case, we wanted to dig a little bit deeper to try to understand what might be causing this difference in participation. And so we looked at a whole bunch of different socioeconomic factors to see which correlated with publication output. And we found that two of the most predictive metrics were national GDP and annual national research and development expenditure. So you can see that the number of papers published by every nation is pretty tightly correlated with their research and development expenditure and their national GDP. In general, wealthier countries invested more in research and development, and this led to more plant science publications. Now, these figures are interesting, and there's some noteworthy cases of individual nations that are publishing, either more or less papers than expected relative to their investment. And I think these would be fascinating follow-up cases where we could go in and do some more detailed digging and try to understand what factors are at play in those countries that are either enhancing or detracting from participation within the plant science publishing field. Just to really drill home this point of affluence being related and highly correlated with publication rates, we can look at how publication rates have changed over time for different groups of nations. So if we look at high income nations, we see that they have always and continue to publish a pretty high number of plant science papers, and there's been a slight increase. Upper middle income nations, so these are some of our emerging economies like Brazil and India, Russia, China, South Africa, have seen more growth, but this is largely attributed to China, which has really exponentially increased the number of plant science papers they've published over the past 20 years. But some of these other emerging economies have also really grown a lot. So we're seeing a lot of growth in these upper middle income nations. Lower middle income nations, I've tried to scale these charts so that the y-axis was more or less even. Lower income nations publish only a fraction of what upper and high income nations publish. And low income nations, combined, all of them together in 2020 published only 140 papers compared to 10,000 in these high income nations. So there's pretty strong correlation between affluence and publishing. And certainly this makes sense. I mean, it's hard to support a robust scientific sector if the country is struggling to meet basic needs and provide services for their population. But it does show that there's a real gap in plant science. Okay, so we also looked at how publication output was related to population density. This is a little bit of a complicated figure, but I'll walk you through it. In general, there's the expectation that productivity will increase with population density and population size. And in general, this is what we found in our data set. So across the globe, more research was published out of cities, basically, compared to small towns. But what we're plotting here, these colors represent how either far above or below the global average an individual city was. And so what you can see is there's a lot of green dots here in the United States, in parts of Europe, and in Australia. And these are locations where they're publishing far more papers than we would expect based on the population density. So this is largely or likely like rural agricultural research institutes. I know I'm at a place like that now in Michigan. It's a relatively rural part of the country, but we publish a ton of plant science papers out of MSU. This makes sense. So plant science is inherently linked to agricultural spaces and to some extent rural spaces. So it does make sense to see a lot of plant science research coming out of rural locations. However, when we look at other parts of the globe, less affluent countries, we see that they're publishing very few, if any, papers out of their rural areas. And in fact, most of the publications are coming out of cities. And so this contributes to the exclusion of rural perspectives of people across much of the globe. And I know that there's a lot of plant science research that is motivated by helping rural communities and improving food security in many of these same regions of the globe, and yet we're not engaging the voices of those people. taken together, yeah, I thought this was a sort of striking stat from the paper. High income nations in Northern America, Europe, and Oceania only account for less than 10% of the world's rural population, but we're publishing more than 65% of plant science papers. So you can see there's certain opinions that are probably dominating the plant science literature and other opinions that are likely excluded. We looked at citation rates on these papers. So we also wanted to understand how research was being used and received. And what we found was when controlling for impact factor of the journals, we found that there were notable differences in the citation rate. So these are citation rates over time. But what you can see, I think, is that authors from China, the United States, Europe, and Oceania were cited nearly twice as many times as authors from the rest of Asia, from Northern Africa and West Asia, from South America, and from Sub-Saharan Africa. I think this is one of those stats that really gave me reason to pause, because citation rates, while they're likely influenced by our network and what's available, it is something that we as individuals have control over. And this suggests that individuals within science are disproportionately citing authors from these affluent regions of the world. I mean, maybe we're just citing our friends, but I think that it's important to recognize that this disparity exists and maybe put a bit more effort individually. All of us can put more effort into finding publications from other authors living in other regions of the world and include those in our citations. Okay, we did a similar analysis to that plant genome paper where we wanted to understand collaboration networks. Because we thought, okay, well, maybe there's really rich collaboration networks that are engaging these authors from other regions of the world. So this big hairball that I'm showing you, the circles represent the number of papers with a corresponding author in that continent, and then the arrows are scaled by the number of co-authors that are in these other continents. Overall, through the whole database, we found that international and intercontinental collaborations were extremely uncommon. Over 70% of the papers in this database only included authors from a single nation. There were 22% that included authors from 2 or more nations, 5% that included authors from 3 or more nations, and less than 1% that included authors from 4 or more nations, despite the fact that over 75% of papers in our database had 4 or more authors. They just tended to all be from the same country and probably all from 1 or 2 institutions within that country. So collaborations were relatively uncommon within this database, and when they did occur, they tended to be between these sort of well-established big plant science producers between Europe, United States, and China. So again, there is room to expand and engage more collaborators across the rest of the globe, and I really believe that this will enrich our science. And I think this is something we can do as individuals, but also as institutions and working groups. And I think that's one of the things that's exciting about ag biodata, is that there really is an effort to try to do this within this working group. Okay, so we kept going. We wanted to look and see if there were more disparities on other axioms of diversity. So we looked at gender dynamics in plant science publishing. And in order to do this, we had to infer the gender of plant science authors based on their first name. Now, this is potentially problematic for a number of reasons. So these algorithms that infer gender based on name perform not as well on certain name types. So Western names, they work pretty well on, but other naming conventions, they don't perform as well. And they also almost certainly misidentify all sorts of people who don't have traditional gender identities. And so we wanted to be clear that we're not presuming to know the true self-identified gender of these authors, but we're measuring the masculinity or femininity of the name itself. And in fact, discrimination and bias can be imposed on names themselves. Imagine a reviewer seeing the name of an author, pegging it as a certain gender, and then invoking some sort of implicit bias, perchance. So all that to say, take these results with a grain of salt. I think across the entire database of 300,000 papers, these patterns are informative and likely true, but we would caution anyone from making detailed claims about the gender of individual authors. Okay, a lot of preamble there, but what we found was that 2/3 of the papers in the database were led by authors with masculine names compared to feminine names. So that's quite a lot. We looked at how this was distributed across the globe, and we find that there's really quite a lot of variation in the gender ratio of these different regions of the world, but a lot of the big producers in plant science, like the United States and China and Western Europe, are extremely male biased. Some regions of the world that had more gender neutral ratios or gender equal ratios are parts of South America and Eastern Europe. And again, I think these would be interesting case studies to follow up and try to understand what are the factors that are at play in these regions of the world that are encouraging or allowing more females to participate. We also looked at citation rates, and similar to the global disparities, we found that despite publishing in similar impact factor journals, females were cited less frequently than males. So again, this just shows that some of these biases are pretty deep in the field. We looked at how gender ratios have changed over time. We thought, well, maybe it was really bad back in 2000, but it's gotten a lot better in 2020. And while we do see some improvement in gender ratios, this improvement has been relatively slow, and all regions of the world lag below 50%, and most are considerably below 50%. Again, I want to point out Latin America, which has maintained a relatively high proportion of females publishing in plant science. And it's not my area of expertise at all. I've never even visited Latin America, but I think it would be really fascinating to try to understand what factors are at play that are enabling females to participate in plant science in these nations. Okay, so this is the last set of analyses, and then I'm going to wrap up. And I'll go through this part kind of quickly because it's very similar to what I showed with the plant genome paper. But here we wanted to conduct a similar analysis to understand which are the most studied species. So in order to get at this, we used natural language processing to recognize species names in the abstract text of these papers. And then we summarize which were the most commonly referenced species. So there's some caveats here. It might not be 100% guaranteed that if the species is mentioned in the abstract that the paper focuses on it. But we're reasonably confident that if you're going to mention a species name in the abstract, it's probably a big part of your paper. There were no big surprises here. Arabidopsis thaliana, the model plant species, is studied in 4 times as many papers as the next most common species, which was wheat, followed by maize, tomato, rice, barley, soybean, apple, tobacco, etc. You'll notice that these are all major agricultural and economically important plants. And in fact, you have to go well below the top 50 species before you come up with anything that's not a sort of major crop. So it's pretty similar to the plant genome database, where we've really prioritized these agriculturally and economically important species. We did a similar analysis looking at over and underrepresentation of studies at an order level, and we found basically the same thing as in plant genomics. So overrepresented groups included Solanaceae, Brassicales, Rosales, Fabeles, Fageles, Poaceae. And underrepresented groups were like Asteraceae and Asparagales, which are super speciose and ecologically diverse plants. So as a field, we're really focused on species that are relevant to humans. And I don't know, it's not necessarily a bad thing, but I think there's a lot of uncatalogued and exciting diversity within wild and biodiverse species that has been possibly overlooked. To that point, we looked at what were the most commonly studied species in each nation from this data set. And I'm just showing you a selection of some of the nations that we looked at. And what I'd like to point out here is that these countries here in the top, these are many of the large producers of plant science literature, and they tended to prioritize sort of obviously, because they're producing most of the research, they follow the general pattern that I showed you on the previous slide, a lot of studies on Arabidopsis, followed by some of these staple and elite crops like maize and soybean and rice and wheat. Once you move on to some of these other nations that have a smaller footprint in the plant science literature and maybe less national affluence, we start to see that there's a really different prioritization of study species. And so this indicates two things to me. One, I think it drives home the idea that priorities may be different in different regions of the globe, and that what we prioritize in North America may not be the same scientific objective that people in other regions of the world have. And secondarily, I think it suggests that if we were to find ways to increase participation on the human level, and get more engagement from these other regions of the world, these underrepresented regions of the world, that we might see a natural diversification of study systems and scientific questions. And I think that that will be hugely beneficial to the field. Okay, so with that, I have no idea how I'm doing on time, but I'm going to leave you guys with just a couple of takeaway messages, and then maybe we can open it up for questions and discussion. So both of these studies identified some major gaps and inequities in plant science. I've argued that these have been driven by historical forces of colonialism, and that they're perpetuated by modern practices involving a lack of collaboration and, in some cases, parachute science. All that being said, I think the really important work here is in developing solutions, and that's why I'm excited to be part of this working group within Ag Biodata, because this is an effort to work towards a solution. In my view, some of the solutions that we can think about include building and supporting international collaboration networks. We can certainly do this as an individual. Go out and engage collaborators from other regions of the world, but we can also do this on more institutionalized levels. Certainly, there's a need for and a justification for working to build global capacity and engagement. I think that this could start with just trying to amplify previously excluded voices and bring more people into the conversation. But there's also a role for creative funding opportunities, for technology transfer, and for, I think importantly, investment in infrastructure. So a lot of times the facilities that are available in some of these less affluent regions of the world are an extreme bottleneck. So it's easier to sequence genomes in the United States because we have a lot of sequencing facilities, for example. Yes, and lastly, this is one of my favorite refrains, but I think there's an incentive, or there's a justification for the need to expand the taxonomic scope of our study systems and to try to catalog the world's biodiversity before it goes extinct. And so with that, I'll just say that I didn't do this all on my own. There's lots of people to thank, and I would like to thank you guys for attending and for your attention, and I'd be happy to take questions or comments.
Well, thank you, Rose. This was a great, really enjoyed it. So thank you so much. So there have been a couple of comments, but anybody that wants to jump in with a question, now is the moment. And I also have some question. So in the chat, well, I think John is suggested a program that, right? Yes. So he's like, he posts.
Hi, everybody. Yeah. Thanks, Henrietta. Yeah, I think, you know, as far as the committee is concerned, I brought this up before, but if those of you don't know, The Academy put out a report late last year on enhancing collaboration among land-grant universities. It was a report requested by the Congress. And right now, they are following up with an implementation report. And a couple of things they address are specifically this: larger, more cooperative studies, directly pulling in underrepresented groups. So, Rosa, you, of course, pegged it exactly. The cycle of disequality, poverty, lack of money, lack of GDP, poverty, that's been there. And so one of my questions has always been, okay, so now what do we do? And you've pegged that. We have to keep getting the word out. And a lot of it, okay, so how do you do this? Okay, so a particular government, a particular administration may decide, okay, it's time to do this and try to, get it through. And this particular administration is doing that. They just announced another program for enhancing workforce in HBCUs and tribal schools. So my point is, I think there's a lot of possibilities here. Ag biodata, was kind of formed partly in order to help do that. So I would just encourage all of you that haven't looked at that report yet, please do take a look. It went through a number of iterations, from a long list of everything that's wrong to a very nice list of what can we do working forward. And take a look at, if you just go to the Board on Ag and Natural Resources of the National Academy, you'll see the information on the ongoing start of the implementation phase, and there'll be, in the next several months, several opportunities for comment. So I think now is the time to really take a active look at that. So thanks, Rosa, for pulling all that together.
Yeah, no, thank you for sharing that report. I actually haven't seen it, so I'm excited to read it. And I would just, sort of echo what you're saying, that I feel like there's a lot of energy right now in the community to talk about these issues and to try to figure out solutions. But of course, it's not as easy as saying, oh, we identified a problem and now we're going to fix it. It's really hard to combat some of these things when they're grounded in like long-term and systemic inequalities, where there just aren't as many resources and opportunities for people in certain parts of the world, even if they want to participate.
Yeah. So like echoing like the inequalities also in terms of technology. So Fortune, she said that, from my experience as a researcher from Sub-Saharan Africa, the cost of sequencing is very high. That has contributed to low research on genomics. I remember when I did my PhD, I literally had a partner with the research institute, and to be able to sequence, it was turned out to be a lack of access to infrastructure and technology. So I guess also that contribute to I have like one, so like I guess like another, I was just thinking, I'm not like a database expert in terms of like, how the information are like processed. So I think if there is anybody here that represent databases and wants to correct me or jump in. But so I was very curious about the inequalities in the citation. So like, that, okay, there, I think like, well, the number of publication are low in like they are higher in Western countries, or like countries that they have like they have more money, also because there are more private investing. So I guess like, for example, United States, there is a lot of contribution from the private system, or like like the growers board, that like I have worked for 3 years for the Walnut Board when I was a postdoc, and they had like a lot of funding towards like the sequencing of the walnut genome and genotyping the breeding program. So I'm not sure if, like, I'm also in Italy, I don't think there is so much investment from the private sector towards like this type of but at the same time, I don't know if like the bias in the citation and knowing about these works, the problem is that sometimes also when you want to create a collaboration, if you don't have a publication, if you don't, or a presentation from scientists at conferences, because maybe they don't have the possibility to travel or like, you know, to be invited, it's also difficult to establish this type of collaboration. And so my question is if there is also kind of bias in how the databases, the literature databases, they process the paper. So let's say, for example, when I want to look for a genome paper on a specific species, if I type that in the researching tool, what kind of paper will pop up first? I don't know if it's based on citation. And so there, the more the paper is cited, of course, people will look at the same paper over and over, and not, maybe there are many other papers that they have been published, but no one knows because they don't pop up in the database. So did you guys like look at that, or is there a way to look at that? Or do you think there is any suggestion about this?
I think that's a really interesting observation. It is not something that we looked at, and we did a lot of sort of broad analyses in this paper, and not as many details. And one of the things that had occurred to me with just understanding the citation rates was more along the lines of what you said of, we might look for papers of people we know or are aware of, or we might try to collaborate with people we've met at conferences. And certainly there's disparities in who can afford to go to conferences. But I hadn't thought about how search engines and publication repositories may be contributing to this. And I know that a lot of, it depends on how you set up your search, right? But a lot of the common tools will by default organize things by citation rates. And so, like you said, a popular paper will become more popular because it's coming up first on everybody's hit. But I'm not a manager of databases, so maybe somebody else in the room can speak to that.
Yeah, Doreen, I was expecting you to jump in as an expert.
I kind of wanted to give a slightly different slant. So my as the links of disparity and things that are going on that you've put up, I think it's clearly true. I mean, as scientists, if we would think about the principal components that are contributing to this, the major one would be what funds the research and who funds the research, right? And what I also want to bring up is perhaps considering that who publishes in the journals that are indexed under these search engines and are part of PubMed are also a bias, because there are a lot of plant journals that actually potentially might be encompassing some of the research from these other countries that may actually be published in their language and not indexed here relative to that. And that's a gap that you're missing. I mean, it's when we started out working in the databases, one of the things that we identified is, especially if you look at an example with China, there are a lot of journals that are in Chinese that are not indexed in here. And so I'm curious whether you've thought about looking at methods of publications that are associated with the countries of origin that you're looking at. And if you were to look at that, because I definitely think publishing in some of the larger journals are the journals that are indexed in PubMed now is a very high rank. And even if you look at the difference between who publishes in an open access and paying for that versus who publishes in something that's not is there. And then I'm just thinking about that as potential a way of looking at ascertainment bias. And then I also would say that when it comes to working in agriculture, some of the people who are very basic people working in agriculture, publications aren't their priority focus. For many of those groups, their priority focus will be getting the germplasm out and getting new varieties out. And even when we look at plant publication registrations in the US, that index isn't even isn't indexed by PubMed. So I think that that even starting with things that are published is starting and creating an ascertainment bias, perhaps associated with, very much biased towards science that's done in these countries that are showing the bias as well. So I'm just bringing these up as things that you may think about when you're looking at what we call publications right now.
Yeah, thank you for those comments. I think that they're both well taken. I would be really interested in expanding on this work to look at some of those other sources of scientific output, right? And I think in particular, the idea of looking at germplasm and varieties and non-published, not so academic outputs would be really worthwhile. And in terms of the journal selection, I think that's a really valid point, and it's something that one of the reviewers raised to us. And we actually did go and expand our journal selection a bit based on that. But it's almost, undoubtedly, we're missing a lot of subject-specific journals, non-English language journals, local journals in different regions of the world. Our justification for that in terms of this paper in particular was that we were focused on the mainstream scientific literature. And if those, not to say those papers and journals aren't valuable, but they're probably less widely read, at least in this field. I think a really important follow-up would be to look at those journals, and if they do show a different pattern than what we found here, amazing. Right? And all the more reason to promote those journals and not reinvent the wheel. But if those journals also show the same pattern that we're finding here, well, I don't know, then we're back to square one. Maybe that's something that we could collaborate on in the future. I mean, I'm not, my expertise is not necessarily in publication systems or in data management, but I think these are worthwhile projects, and, or if you guys want to take these ideas and run with it, be my guest.
So I had also comments on this, but maybe I will take the question into chat, and because I think we are running out of time, but we can talk more about this. It's very interesting, I think. So the question is really wonderful and stimulating presentation. Thank you. You mentioned being dissatisfied with the rate of increase of female authors. Would you be willing to speculate on how that rate of increase might compare with changes in participation of authors of minoritized races? It's hard to imagine a way to track that rate. In our current moment in the United States, it seems important.
Yeah, great question. So we did not address race directly in this paper because we had, like you've implied here, we had no way to ascertain those data. I think this is something that could be looked at if we took a different angle, and perhaps we're looking at participation in science based on enrollment in degrees and programs where they do track some of those data. I'm shooting from the hip here, but I'd be willing to speculate that there's even slower increase in participation of minoritized races compared to gender. I mean, if we look sort of historically across how women's rights have moved forward compared to racial equality, I think we generally find more progress on gender issues. So my gut feeling would be that the picture is even bleaker if you look at the racial component. But I didn't know exactly how to get at that. I think that would be a worthwhile project as well. So thank you for these great questions and suggestions. I wish we could just keep chatting.
Yes.
People have places to go.
Out of time, and actually Doreen. So I think another perspective, just we leave these since we are part of biodata, there are a lot of database member. Maybe we should conclude with a question like how the database can actually help these researchers to get out their data, even if they are not supported. Sometimes they are small projects that they cannot land in a very big journal, or they cannot afford to publish for open access. But I'm just wondering if the database can actually accept their data and get them citable, and so they get visibility. So that will be another way that maybe we can be more inclusive for these communities, I guess. Well, thank you so much, Rose. It was very great. So I will post the recording on our YouTube channel. And thank you so much for who joined. Some people already left, but yeah, thank you so much, and have a nice week, you all.
Thanks, everybody.
Bye-bye.